Thank you very much, Jinan. Professor Joseph Wang is a distinguished professor and chair of the Department of Nanoengineering at the University of California, San Diego. I encourage you to read his full and extraordinary bio on our website, including links to his 1,060 papers. Welcome, Professor Wang.
Thank you, Lisa. Sleeping mode. Sleeping. What happened here? Huh? Oh. Okay. Switch the screen here. Thank you, Lisa, and thank you, Jinan, for hosting us. Pleasure to be here. So coming down from UC San Diego in La Jolla Cove, and this is our skin, electronic skin, the UC system printed on the skin. And we have also a center, wearable center, CWS. So as we saw today, the digital revolution, arrival of smartphones, there are a lot of activity on on-body sensing. But what is missing is the chemistry. So there are a lot of devices for monitoring activity, vital sign, as we saw before. But the major gap is the lack of chemical information. Jinan touched at the end. We mentioned we'll have some more talk about dopamine, serotonin today. But overall, to get comprehensive information about the health, fitness, performance, stress, we would like to have 24/7 chemical information of key biomarkers. So this is what we have now in the market on my iWatch and so on. So we can measure physical parameter, electrical parameters in untouched nicely. But what is missing is the chemical. So we have a little bit discussion today. But this is what my lab is focusing for the last decade, to introduce wearable chemical sensor, biosensor, for monitoring, non-invasive monitoring of key biomarker. So this is the opportunity, but at the same time, there is a key challenge why it was not available. And the challenge is the stability, the issue of biofouling or passivation of this chemical sensor, biosensor, selectivity due to coexisting electroactive interferences, and changing uncontrolled condition, pH, temperature. All of these are major challenges why we don't have yet a continuous glucose sensor despite the huge market. So we are aiming at this parameter. We like electrochemical detection, like what you see in the glucose market, because of many attractive property, suitability for wearable device, such as high performance, miniaturization, low power requirement, low cost microfabrication, and easy to use. So we'll focus on printable electrochemical device. If you visit my lab, you're welcome by these many cans with many, many sensors from head to toe, on textile, on the skin, in the mouth, in the socks and the glove, and so on. So we'll talk about this in the next 40 minutes. Primarily, we'll talk about, following Jinan, electronic skin, and then we will move to a textile-based sensor. We'll talk about in-mouse monitoring and microneedle for minimally invasive monitoring. So all about chemical sensing of metabolite, electrolyte, protein, biomarker, non-invasively or minimally invasively. So this is what the target biofluids that we are aiming for these: sweat, saliva, tears, ISF, monitor the surrounding, and variety of biomarker ranging from, again, metabolite like glucose, lactate, electrolyte, sodium, potassium, pH, calcium, stress marker, cortisol. We'll talk about neurotransmitter. We'll talk about dopamine and serotonin, Lot of disease marker. We're pushing under the skin for cytokines monitoring. We also have a lot of defense application for nerve agent and environmental and so on. So the goal is to have 24/7 continuous monitor of health, performance, and safety. So as I mentioned, we'll talk about electrochemical sensor, especially printable sensor. This is screen printing, has been widely used for over 2 decades in the glucose market. All the glucose strips in the 80s and 90s are based on printed electrode like this. And we will adapt them for our skin and for our textile-based device. So typically in our lab, we have a printing on stencil with a pattern of the electrode, this sensor array for different injury, TBI, soft tissue injury, liver damage. And we are transferring the pattern, putting the ink, transfer it to a textile. This is Gore-Tex textile. So the same 8 electrode array, and these are the carbon part, and we have the silver contact, and then we have the insulator transferred directly onto a flexible substrate or textile. So as Zinan mentioned, we have to merge this is the rigidity of electronic with the softness of our body. This is from San Diego Zoo. If you visit us, you see these flamingos. They are very stretchable, very soft. But unfortunately, this battery or glass PHLs are all rigid, bulky, and planar. So we need to merge the two, and we will see, like Zinan, how to develop stretchable and self-healing materials. So the goal is to, again, stretch our imagination, stretch our electronic skin by developing this stretchable, printable device that not only can stretch, can bend, can twist, and self-repair by using new material, ink material, which are stress-enduring, and new structure like serpentine that can stretch, and so on. So we combine these 2 dimensions of stretchability. One is the structure. The serpentine allows you 1 dimension up to 200% stretching. And then with the material, we go all the way to 500–600%. Using the ink formulation, which is stress-enduring with elastomer, such as polyurethane or Ecoflex, we can push the limit with these different binders, which are elastomeric. So, So as you can see, we use this design of the stencil with the serpentine, and the ink are stress-enduring, so we can push it and we can stress and stretch the device 200–300%. We can twist it so without compromising the electrochemical performance, electrochemical property, and so on. So this is not only for sensing, we use it for energy device, for biofuel cell, for supercapacitor, and so on. For environmental sensing, we put them on balloon. This is for security and environmental. And again, you inflate the balloon, we'll have the electronic here, and we can monitor the surrounding. As Zinan mentioned, we not only stretch. If you stretch too much, you can break, and we're also working on self-healing printable material. In this case, we use this magnetic self-healing, incorporated this particle into the ink, and this allows you to repair big crack up to 3–4 millimeters repetitively at the same location in 5 seconds, multiple times. So it's very attractive for wearable application. So with all this capability, now we can go to our chemical sensing on the skin, in the mouse, or under the skin. So again, monitor chemical marker, biomarker, metabolite, electrolyte, protein, and so on. So as Zinan showed nicely, our skin is the gateway for our biochemistry. And with the skin monitoring, we can monitor both sweat, also the ISF, subcutaneous monitoring, where there is more information due to biomarker closer to the blood, like cytokine, and so on. So we want to develop this lab on a chip, both for sweat and ISF monitoring. So early work, 10 years ago, we went to the beach in San Diego. We got some nice tattoo paper, and we print our sensor on a temporary tattoo. This is low cost, printing on a low cost temporary tattoo paper, and then easily transfer it to the skin. We make it flower-like shape. But these are the three electrodes, the working, counter, and reference electrode. So they combine the elasticity of the temporary tattoo with extractive electrochemical performance based on the ink formulation. So readily transfer it to the surface by peeling off the protective layer, take 5-10 seconds. This is cost of less than a dime, and you can transfer it and reuse it or dispose it very quickly. So we can apply it two ways. If you want to measure the sweat, you can face it to the skin. If you want to measure the surrounding environment or security application, you can face it outside. Again, you can twist it, you can stretch it as needed. So this combines, again, a nice electrochemistry of our novel ink material with epidermal sensing, flexible device, and screen printing to do continuous biomarker monitoring, health and performance. So our early work 8 years ago, we started 2011, actually, published later on, was the first biosensor on the skin for lactate monitoring. Lactate is extremely important for performance and muscle fatigue. So we immobilized, this is my nano-engineering department. So these are my three electrodes working reference and counter. We immobilize the enzyme lactate oxidase, which is highly selective on the working electrode. We start to exercise, generate sweat. You see the lactate profile, which is a measure of the fitness level. Without the enzyme, you see no response. Nice performance. This is in vitro calibration, nice calibration at 1 to 20 millimolar. These are the sweat lactate concentration. So extremely important for fitness and performance monitoring. Later on, we combine it with ECG. This is the first example, coupling biosensing with physical parameter like ECG on the same patch. So it's all printed, the ECG electrode and the biosensing, glucose and lactate. And you can run simultaneously the ECG and the lactate or glucose without any crosstalk. These are two different subjects. The electronic is combined both for the electrochemical and ECG, all in the same patch. So from lactate, we move to glucose. Glucose, obviously, is the biggest market need for diabetic monitoring. And again, there is a major dream to have non-invasive, continuous 24/7 monitor of glucose. And again, with this device, you can readily do ISF monitoring. We'll talk about reverse iontophoretic, extracting the glucose, which is very correlated to blood glucose, and more recently to sweat glucose. So 2015, we show this tattoo, which combined reverse iontophoretic, extraction of glucose to the skin. The same tattoo both extract the ISF and then measure the glucose. Again, before the meal and after the meal. This is single-use device, again, a dime cost, and it readily placed on the surface. You can see it in the museum in Vienna, the City of the Future display in Austria. So again, this do both the iontophoretic extraction as well as the biosensing with glucose oxidase. We'll put the electronic here. The electronic is reusable. And this is on the tattoo paper. And very rapidly, you can see the video transfer it to the skin, peeling the protective layer. And this is slow motion. Usually, it will take 5 seconds. And it's mostly for single use to replace the finger stick. So it's the same cost like a strip, but you don't do any finger stick blood. So again, this is a slow motion, and we'll place the electronic here, which is reusable. So with this, you can obtain, again, before the meal, after the meal. It takes a total of 2 minutes, 90 seconds of iontophoretic extraction, another 30 seconds of the detection, nice correlation to blood glucose meter. So it's a single use. You can do it 3-4 times a day. And again, this is a conformal electronic, wireless, Bluetooth, which should do the amperometric detection, have the energy, control the device, transmission to a smartphone or a laptop, and so on. So after eating, we're also drinking. Hopefully tonight we'll have some wine beer, and we will test the alcohol level. Alcohol actually is easier. This is stimulated sweat where we use with pilocarpine the same tattoo, but now instead of extraction, the ISF, we stimulate the sweat, and we bring the sweat in 1 minute to the surface where you have the alcohol oxidase. Again, the same conformal electronic with wireless. And here we have the enzyme alcohol oxidase on this printed Prussian blue transducer. And you can see eventually it will tell you how drunk you are within 2 minutes. This is correlation to blood alcohol level. And this is without drinking. As you see, no change. All the controls. So again, similar to the glucose. And later on, we combine it recently in what we call the glucol device that can measure both glucose and alcohol. So you can eat and drink and measure both parameters. This is unique because this tattoo, panda-shaped tattoo, actually measures 2 different biofluid, sweat here and ISF here. So when you apply the iontophoretic with the pilocarpine, you stimulate the sweat to measure the alcohol on this, where you have alcohol oxidase. At the same time, glucose is transported with the ISF to the second electrode, and you get your glucose in ISF. So again, alcohol, glucose, no eating, no drinking, no response. You can twist it, you can stretch it. It takes about 3 minutes to have combined. So the extraction about 2 minutes, another 2 minutes total to get the glucose and alcohol readout. So Zinan mentioned different accessory in the future. So we put our, what is this, Dolce Gabbana glasses. You can see, because I have nice big frame to put my electronic, not because I'm promoting DC, but basically I can put all my electronic, and this can do sweat here, where you have on the pad, you put both potentiometric sensor like potassium and amperometric for glucose or lactate. And you can see simultaneously electrolyte and metabolite contacting nicely the skin. This is calibration for potassium, and this is for glucose and lactate, all wireless, transmitted, controlled by these 2 electronic boards. And the way we do, we peel these are reusable or disposable sensor to place here, one for potassium or sodium with the receptor, the ionophore. This is the glucose sensor with glucose oxidase or lactate oxidase. So these are, again, printed, easily transferred. And you can measure simultaneously. This is potassium and lactate in sweat. So we mentioned potassium. This is a different group of electrochemical device known as potentiometric. This is the oldest group. If you remember, the glass pH electrode is over a century, 100 years ago. We have this classical glass with inner HCl solution. So obviously, this is not suitable for wearable application. You have a very strong 1 molar HCl in glass. So we want to transfer it to a flexible potentiometric sensor. This is what we call solid contact ion selective electrode. This is the classical ion selective electrode, IEC, and this is solid contact with printing the various transducer, and we put the receptor, let's say for ammonium or pH, on the working electrode transducer. So we started with the pH, I think 7 years ago. This is my Smiley pH sensor. Again, taking this rigid glass electrode, convert it to a flexible Smiley pH on a tattoo paper. We print, these are 4 different sensors where the eyes are the 2 electrodes, reference and working electrode. We put the receptor, the ionophore for the electrolyte, contact through the ears, and we have a Smiley pH sensor on the skin. And as you can see, we can subject it to many, many repeated mechanical strains. These are 50 bending. After 50 bending like this, it's still smiling. You see, still smiling before and still responding. More important, the performance is not compromised despite of this severe stress. So you see, nice smile and good performance. This is a sodium flower-like sensor with another ionophore for sodium. And this is a different design with a little channel so sweat can flow, and you can push it to the limit, and you see This is 100 repeated stretching. Every 20 stretching, you see the calibration. 100 bending, every 20 bending. And again, carryover and calibration, what you expect from good ion selective electrode. Nutrient and trace metal in sweat. The zinc is very important. We work with DSM, is major supplier of nutrient for sweat. Zinc, for example, or vitamin in sweat. This technique called stripping voltammetry, we can measure parts per billion level. You have a short accumulation, 1 minute accumulation, and the stripping, the measurement sweat on a bismuth a tattoo electrode, very reproducible, at the low ppm parts per billion level. More recently, we moved to fluidic, combining the electrochemical sensor with epidermal microfluidics that allow continuous transport of the sweat. So we have sampling of sweat, multiple inlet over the detector. This is the electronic, and then the sweat is discarded. So we have continuous supply. We minimize carryover. We minimize mixing of the sweat, old sweat and new sweat, and it's continuously flowing. This is the design on PDMS. We have 4 inlet to have good, efficient sweat flow. Again, you can twist it and bend it, and you can see in the video, this is how we make it. So this, we put a dye. In 10 seconds, we are filling the detector compartment. We remove it here, so the sweat is pushing my dye. And this allows you to measure sweat lactate, sweat glucose. Here we have sweat lactate during exercise again. So again, once you generate the sweat, you're reaching the detector, you see the profile for different subjects. And this is for electrolytes, sodium and potassium. We have the 2 electrodes. And again, sweat is pushing the dye and over the 2 ion selective electrodes. So the inlet and its flow. The outlet is opposite side. So this was a epidermal monitor on the skin directly. Is a tattoo or a microfluidic? Sweat is a textile, is, as Inan mentioned, is very attractive for future application. You can go all the way. There's plenty of sweat at the bottom. This is in our socks. We can detect a lot of parameters in sweat, but all the way up, both on textile, shirt, underwear, and we started it again 10 years ago. And more recently, we use it for protecting soldiers. This is their agent detection for the army, for military application. And, but the early work 10 years ago was my smart underwear, where we printed on the elastic vest, this contacting the skin. This responds for uric acid in sweat using these are big 1 millimeter electrode on the underwear. Again, I show you this textile for array of injury, different injury, traumatic brain injury, a liver injury, soft tissue injury on a Gore-Tex using this stencil. And we can use it to measure vapor of explosive, also for protecting our soldier TNT and DNT at the ppm level. And again, this again can be stretched, multi-axial stretching and twisting to the limit. We push it to the limit. We even wash it multiple times. You can see it survived this harsh condition of the washing. And again, this is a different textile sensor for electrolytes. This is sodium and potassium, so we have the 2 ion selective electrode. We replace, normally they use PVC. We replace the PVC with elastomer, with polyurethane. They have the serpentine design. So again, we combine the structures that stretch, the serpentine, along with the ink, with nanotube and polyurethane. And again, you can stretch it to 300%, the sodium, potassium, with a joint reference electrode. So again, instead of normal PVC, we now have polyurethane to have the ionophore receptor for this potassium and sodium. As I mentioned, we work with DITRA on defense application, and this is for nerve agent, organophosphorus hydrolase. So the nerve agent will be converted by the enzyme to a detectable nitrophenol. You can do it on the skin or textile to protect. These are increasing level of the vapor of the nerve agent. Here it's pesticide. We don't do it on the skin. This is a mannequin for safety application. But see, increasing level of the nerve agent vapor, the increasing peak of the nitrophenol product. So the enzyme is immobilized and then same time generate the nitrophenol, which is electroactive. And you scan the voltage, you get a nice peak at 0.8. We worked with the Navy over 10 years ago on underwater explosive detection and pollution in San Diego Bay, like copper. So we print the text sensor directly on the wetsuit, on the wetsuit of the neoprene rubber of the wetsuit. Again, all the electronic, you get the elevated level, you get the red light and warning, both for pollution or for explosive. This replaced the dolphin. And Navy in San Diego used live dolphin before, so this is more to develop it on the wetsuit directly without the need for dolphin, both security and environmental application. So this is like copper, parts per billion of copper in San Diego Bay, coming from the paint, protective paint of the Navy boat. So there is elevated level of copper and tin. So again, this is the wetsuit sensor, parts per billion, very stable and nice calibration. Bandage, we do a lot for wound healing, wound management. Again, the same pH sensor we talked before with polyaniline. Again, putting on a bandage. These are 100 repeated bending like this. Respond every 20 bending of the bandage, and then you see the lack of carryover effect. And that's all combined with NFC or wireless communication. This bandage is for uric acid. Uric acid is an important parameter for the wound healing and management. We immobilize the enzyme uricase. Uricase convert the uric acid to peroxide eventually, which is detected by the Prussian blue transducer. And more recently, we published last year, this is the first bandage for cancer screening. So in the case of melanoma mole, you have elevated level of the enzyme tyrosinase expressed inside the mole. So we developed a bandage where we immobilize the substrate, the catechol substrate, and then in the presence of tyrosinase or melanoma, you'll have it will convert the catechol to benzoquinone product, which is detected electrochemically. So this is for quick screening when you go to your annual or dermatologist. In 1 minute, instead of taking the biopsy, you can measure it rapidly. And in 80% of melanoma, you have the enzyme tyrosinase expressed at higher level. Now, as I mentioned, we do a lot of security application, chemistry touch, also for robotic application. We're developing gloves that the robot can do security and also daily operation. But we started the early work with explosive residue 10 years ago, gunshot residue 8 years ago, more recently with DTRA nerve agent. Now we work on opioid and also spices, as you'll see in the next 5 slides. So this is the nerve agent sensor when we immobilize the OPH enzyme. So again, we print the serpentine so we can stretch the glove when you place it. And you have 2 fingers. One is the collection finger to swipe the surface. Let's say you have pesticide on your tomato. You swipe it. Also, you can measure the pressure. We also have pressure sensor capacitor. You can do swiping and measure both the chemistry and the pressure, but also swipe for the military different vehicle with nerve agent. And then you have ionic liquid. You close the 2 fingers together. You have the electronic on the ring, and you can measure the phenol product of the nerve agent. So this is, again, OPH immobilized here. This is collection, and the OPH give you the phenol product, highly stretchable. Early work we did for cocaine drug. We do opioid now. So the nice thing when you get this street sample from Colombia, you don't have only cocaine. You also have your caffeine, thiophylline. And this gives you much nicer signature for our electrochemical detection. Cocaine is a small one, but all these editing cutting agent, lidocaine or benzocaine, acetaminophen and caffeine give you much more information to get the chemical signature. We just published a paper on opioid, on fentanyl, using this approach. And this is also the latest robotic skin. We work with Honda from Japan. This is Asimo. So when I went to Tokyo, Asimo gave me the coffee, but he only have pressure sensor to open the thermos. He don't have chemical. He cannot tell me if there is caffeine or sugar. So we developed for Honda this chemistry, measuring different spices. Every finger has either sour or caffeine or sweet or spiciness. Here is the paprika. And we can measure unique signature for testing food by the robot. So again, this is for measuring. So this Asimo gave me the coffee. But same time, we can measure if it's caffeinated. These are differently coffee, tea, and this is decaffeinated. So this unique signature of caffeine. And again, without caffeine, you don't see it. So ultimately, we are developing these lab-on gloves. We have, how many? 10 fingers we have usually, so we can measure many, many parameters for robotic application, for forensic application. And again, actually, we need one finger for swiping. So we're losing one finger. The other 9 fingers are for different chemistry. And again, all the electronics on a wristband. We also use other accessories like this diamond ring. On the diamond ring, we put, again, for DTRA, the nerve agent sensing and the explosive. So we have 2 sensors, one for explosive residue and one, again, for the nerve agent with OPH. This is the nitrophenol product. And all the electronic is embedded inside the ring. You can see nicely how the electronic, we spray the nerve agent or the explosive residue in all wireless transmitted, and it's all the 2 sensors. We're working now on opioid, we work on THC, on cannabis now for forensic application. So this was on the skin. On textile, we move under the skin. We love microneedle. These are minimally invasive. We have, these are what, 12 individually addressable microneedle for multi-parameters. We can measure 12 multiple biomarker individually addressed. Put glucose, lactate. We measure now insulin, working on cytokine, cortisol. So I have a lot of security defense application using microneedle, alcohol we showed before. So again, this is the way we have with the electronic. This is the 12 or 9 sensor array. This is how it looks. These are 3 sensors. And again, this is the early work 8 years ago. We did glucose and lactate. And you can see we're filling this hollow microneedle with the paste containing the enzyme, glucose oxidase, or lactate. You see nice calibration. This in vitro, very selective. These are the common interferences, uric acid, cysteine, acetaminophen. And then we have a spin-off company, BioLink, commercializing it for diabetic monitoring. This is the alcohol sensor with the alcohol oxidase immobilized on the microneedle, again, for monitoring alcohol abuse. We did, again, the melanoma sensor, we monitor tyrosinase. But the ultimate goal is to combine it with drug delivery. Since we have 12 microneedle, we can have 6 of them to monitor different metabolites or biomarkers. But then we can have different drugs that can be open and closed. These are conducting polymer actuator like polypyrrole, that the response of the sensor, if we have logic gate approach, if we have AND gate with 111, it will activate the delivery of the drug in an optimal fashion. So obviously glucose and insulin, we are working L-DOPA for Parkinson's disease and other closed loop application. So in this case, we show like, here we show that we can deliver different dyes or solution from different microneedle. So again, here both of these are closed. Here we open the right one with the conducting polymer. Here we open both of them. Here we close the right one, open the left one. So it's very nice control from this microneedle. A nice video in this paper. But so overall, we show this approach of logic control, therapeutic and intervention. This is cardiac monitoring. We have 2 biomarkers, LDH and lactate. Only when both of them are present in the end gate 1-1, you can have the therapeutic intervention. Lactate alone is not enough. LDH alone is not enough. Only when both of them are present, impart the therapeutic intervention. We're also developing self-power sensor. This is biofuel cell microneedle, where the anode is a glucose-based anode, and this is platinum oxygen cathode, where the power is proportional to the glucose fuel. So as you see, the power increase with the fuel concentration, so you don't need external battery. Basically, the self-power biofuel cell sensor, and we are working on this extensively. We combine it with supercapacitor, with tribo, energy harvesting, with batteries, and so on. This work of Jiang is sitting here, and she did develop this mouse guard 4 years ago. And again, we have a spin-off company, MouseSend, that continuous saliva monitoring. Again, is wireless transmission with Patrick Mercier, my electrical engineer, my co-director. So the early work of Jiang, we use lactate monitoring. Saliva lactate is important for performance and stress. So immobilize lactate oxidase, nice response. Uric acid for gut disease. So here we immobilize uric acid. And again, wirelessly transmitting the data. So again, uric acid give you peroxide, and you measure saliva uric acid. We're expanding it to other biomarkers. This is a MouseSend spin-off company in Carlsbad, California. And this is Jiang is here. So yeah, I think this video will show you placing this in vitro. You place the saliva, spike saliva. You see, maybe it will take too long. Yeah. But basically, the video will show you. Let's see, Jiang, put it here. Okay, so in 1 minute, you see nice response to increasing level of uric acid, 30 seconds of reaction, and then here you see the uric acid signal transmitted. And now we are integrating everything. We are sealing everything for in vivo studies. So all the electronic is being sealed now completely. We do leaching study, toxicity study to have the first human subject. So this was a NextFlex defense project with the ePark, with the Park and other commercial partner. But you can see the way it looks now, completely sealed, and there is wireless recharging of this. So in conclusion, I hope that I gave you a flavor that we are focusing on chemistry, 24/7 monitoring of chemical biomarker in different biofluids, sweat, ISF, saliva, tears. And hopefully these advances will lead for new opportunity for healthcare, for fitness, but also we do a lot of defense applications. Still, we need a lot of large-scale evaluation, critical validation, like in the case of glucose, but we are on the way into this continuous non-invasive, minimally invasive chemical sensing. So again, let me take my co-worker. Here is Jiang again, and there is Jinjin. This is, Jinjin is another one here. So also a group of 45, 50 people. They're doing the job, a lot of postdoc, visiting scholars. They did all the work. Again, these are the people who work on the wearable sensor on this project. Emma is in Chicago with John Roger. Josh is the BioLink CEO. Jiang is here. Many more, and funding for different government and other company, my collaborator. And thank you all for your attention. Thank you very much.
They're overwhelmed.
Please, yeah.
Thank you very much for a great presentation. In terms of time, as a function of time, continuity of the measurement and sensing, can you comment on that and how the technology evolving on that?
So it depends on the specific technique. Glucose, lactate, amperometric, you can measure continuously. Potentiometric, you measure continuously. But many protein biomarkers, we mentioned cortisol, they need some incubation time, where depending on concentration, many times is nanomolar, even picomolar. We work on insulin, which is very challenging in the picomolar. They need 10, 15 minutes incubation to go to this low level. And again, there are challenges, immunoassay, you need to do sandwich type, bring the enzyme, bring the substrate. So that's why we have all these fluidics that eventually we can mix solution and do it on the skin. But it's very challenging. But a lot of these you'll see work by hand later on. We talk about serotonin. This is continuous monitor. People put electrode in the brain, and this can be done continuously. So depend on the specific technique, yeah. Zinan, yeah.
Joe, beautiful work.
Thank you.
I wonder how much drift do you observe for longer-term wear, and also in terms of selectivity. For example, you showed the panel of different drugs that may be detected or may be in the mixture.
Sure.
How do you tell which one is which?
Exactly. And this has been the challenge. Let's say the glucose market. You have both challenge of biofouling. So even you have device by Medtronic or Dexcom, they last for 7-10 days. Abbott now have device for 2 weeks because of biofouling. So we work a lot of protective layer, both to impart selectivity and stability. So selectivity, you'll see interference of ascorbic acid, uric acid, but you can use electrostatic repulsion coating which are charged like Nephion. They will expel the negatively charged anionics. So the multi-layer of, there's a lot of sophistication on this. It's not only the transducer, so this requires getting rid. Saliva is very complex. You need to protect all the protein in saliva. Sweat is simpler. ISF is simpler. Tear is a little simpler. So there are a lot of challenges. That's why you don't have a glucose watch yet on the iPhone and so on. But Apple is working hard on this, Samsung and so on.
So is there, go ahead.
Yeah,
Hi, Joe. You know I love the electrochemistry.
For sure. Yeah.
I was wondering if you could say just a couple of words about the relationship between some of the things that you're measuring in sweat and what we would see, for instance, in a blood measurement. What's the correspondence or the challenges?
Yeah. So let's say glucose. Everybody dreams to have your glucose sweat, non-invasive. There are mixed reports in the literature. So a group of Kim in Korea shows some correlation, but not large-scale validation. You need a new paper in Analchem last week from Karayakin showed nice correlation. We observed some nice. We still prefer the ISF. ISF has been established as reverse iontophoretic, so you have better correlation. This is what the commercial glucose, the Dexcom and Medtronic, they use them in a subcutaneous monitor, which is well correlated, maybe 10-minute delay compared to blood glucose. So it differs from analyte to analyte. Sweat alcohol is good, sweat lactate is good, but glucose is still mixed. Saliva, uric acid, lactate, cortisol is well known to correlate to blood, to stress, and so on. We cannot detect your neurotransmitter. The concentration are too low, but we are working on L-DOPA for Parkinson. L-DOPA, the level are 10 micromolar for Parkinson patient, and this is quite nice for closed loop because L-DOPA a very narrow therapeutic range. So we're working on microneedle that you can have a closed loop adjusted, yeah.
So again, nice talk.
Thank you.
Tour de force and electrochemical stuff. But the one question I have is, what is the time lag between when I see a certain glucose level in the blood, say, and I can pick it up in the sweat?
So as I mentioned, in ISF, like this tattoo, this is about 10 minutes delay between blood and ISF. In sweat, it may be at least 20 minutes, but even the correlation is not established. Even saliva sweat, saliva glucose or tear, that's why Google have problem with the project in tear. So there are delay, and even the saliva glucose is not, sometimes it depends on the patient. It's correlated, sometimes not. ISF is the most established. There more and more work on glucose. But so glucose is the biggest market, but many other parameters I mentioned, alcohol in sweat, lactate in sweat, they're all correlated well and very useful information.
But in some aspects, you know, the time lag is much more critical than other aspects, right? I mean, if alcohol content, you could say, well, there's a 10-minute delay. All right, but you know, for diabetics, 20-minute delay in knowing that you're gone hyperglycemic could be pretty important.
Sure, no.
But what I'm wondering is, so I expect that time delay, but are there efforts in saying, is there something we can do to shorten that gap by whatever means, electrochemical or chemical? Sure. Is there a way of saying, look, 20 minutes is not enough, I need to bring it down to
maybe
a minute?
5, 10, yeah. So we have like the microneedle, we have a spin-off company, BioLink, and they do the stage 2 clinical validation. They're getting very promising results with microneedle with minimal delay. But also you have 3 products in the market by Abbott, Medtronic, and Dexcom. And they are FDA approved, and you even don't need calibration anymore. And
yeah, so maybe I should have been clearer. I meant in non-invasive testing, right? The microneedles, you could say,
the minimally invasive.
Minimally invasive, but yeah, ideally, if I want to say, well, I don't even want minimally, I want non-invasive, continuous, then is there some effort to say, can I reduce this without being invasive, even if it's minimal?
So if you know, 20 years ago, it was a company here, Cygnus, in Redwood City, yeah? I was in consulting for them. The glucose watch was the first wearable device in the late 90s, and they got FDA approval. The problem was not the correlation, but more irritation of the skin. They use this iontophoretic, and some people complain about skin irritation. But they got a very nice correlation to blood glucose. They got measurement, 4 measurements per hour.
Without the time gap? Without the time lag?
Oh, yeah, minimal. Yeah, it was all approved by FDA. This was the first wearable. I still have it in my office. Cygnus, the glucose watch. The only problem was irritation of the skin and variation of different people with different skin. But FDA approved it. It was 2-3 years in the market until the people complain about irritation. And, yeah. Sure.
So could you expound a little bit further on how you're, with your sweat sensor, how you're getting out the old sweat and getting a new sweat to test for accuracy purposes?
That's why we in John Rogers' team also, they use microfluidic now. Microfluidic, the sweat glands are serving as the pumps. They're pushing the sweat over the detector and then removing the old sweat. You have new sweat all the time. So the Alta 2 was single use, not for continuous. But if you want continuous, the way John Rogers and now we do is we using fluidic. The glands actually serving as a pump, transporting the sweat over the detector, and you have a removal of all sweat. Yeah.
But there's no mixture there between the old sweat and the new
No, the fluidic is not. A very narrow channel, it's just flowing over like a lab on the skin, basically. Yeah.
So off of that question, how long can you keep reusing the same sample or device, given that you are under continuous measurement?
Yeah.
Calibration, whatever you
Sure, yeah, no, it's the same issue, biofouling, but as long as, yeah, and it's not only with exercise, you can also stimulate the sweat using the pilocarpine using this device. But these are very low cost, coupled with the electrochemical is low cost, so it's usually for daily application. And John Rogers showed recently, few days, 2-3 days, etc. Thank you. Okay, thank you very much.
Thank you very much. Speaking of John Rogers, he and Ruz Ghaffari came out with Epicor Biosystems. I don't know if you've been reading about it the last few days, but it's continuous sweat monitoring.
Thank you, Lisa.
Thank you very much. We're running a bit behind, and we've canceled the break, so we're going to. Sorry. So, but the food's going to be in the back of the room. So feel free to grab a coffee anytime you'd like. Our next speaker is John Madison, Chief Medical Information Officer and Assistant Medical Director at Kaiser Permanente.